handling blood group serology. So today we want to look at some of the reactions that occur between the red cell antigens and the antibodies that we find in the plasma, in the plasma of the blood. So we want to look at the type of interactions between the red cell antigens and the antibodies in our plasma. So that's what we are going to do basically today. So we have some basic antigen antibody reaction interactions. Okay, that's what we are going to discuss. We have the first of them, we have what we call agglutination of red blood cells. Okay, and that has to do with the clumping of red blood cells due to antibody coating. Okay, we know that the red blood cells are coated like you see in this diagram here. You can see in these diagrams here, the red blood cells are already has their antigenic structures outside of their cell membranes. Okay, so the main reaction we look for in blood banking, that is the agglutination. So it causes red cell clumping due to antibody coating. You know, the red blood cells have their antigens around their cell membranes, and the antibody, if there's a corresponding antibody in the plasma that attaches to them, it coats the red blood cells. Okay, it coats the red blood cells, and this agglutination occurs in two steps. The first one is the coating of the cells, that is the sensitization of the cells. Okay, it's carried out by the antibody specificity. Okay, it's being affected by the antibody specificity, electrostatic red blood cell, temperature, amount of antigen antibody present. These are the factors that affect agglutination. Also, low ionic strength saline decreases repulsive charges between red blood cells and tend to enhance cold antibody and autoantibody. Polyethylene glycol PEG removes water and tend to enhance warm antibodies. So in agglutination, which is a type of red blood cell interaction that happens in the body or outside of the body, okay, it occurs as a result of the interaction between the red blood cell antigens, surface of the cells, and the antibodies on the plasma. If there's a corresponding antibody, it attaches to the red cell antigen and causes what? Sensitization. Okay, there are some factors that enhance, encourage, or increase the occurrence of agglutination. Like we said, it's affected by antibody specificity. Okay, if assuming somebody is A blood group and in the body has the corresponding anti-B. Okay, but if mistakenly, maybe by transfusion errors, the person is being transfused with blood group, let's say blood group B, which has anti-A's in their serum. So the anti-A will attack the A antigens in the patient's red cells and lead to what we call agglutination. That's what we mean by antibody specificity. The antibody and the antigen has to be specific for the reaction to occur. Also, electrostatic red blood cell charge, temperature, amount of antigen and antibody affects this process of agglutination. Low ionic strength saline will decrease the repulsive charges. There's what we call repulsion, like charges repel. So the repulsive charges between the red cell antigen, this low strength ionic saline, if the reaction is being carried out in such an environment, we reduce this repulsive charge and makes the red blood cell to come together to enhance the reactions. And it normally encourages what we call the cold antibodies. Okay, we also have PEG, which removes water and tends to enhance the agglutination of the attachment of warm antibodies to the antigens. So if you see this diagram here, there are some antibodies we call the complete antibodies, mostly IgM, because the IgM is a pentamer. Okay, it's a pentamer, so it attracts cells more rapidly and comprehensively complete reaction, while the incomplete antibody, the IgG, is just a monomer, so it doesn't have that capacity like the IgM to bring cells together by binding. So agglutination is a type of interaction we see in red blood cells. Okay. So we also have the formation of bridges. We have the formation of bridges, lattice structure formed by antibodies and red blood cells. Okay, the IgG is not good at that. It's just a monomer. You can see the IgG diagram here. One antibody arm must attach to one cell and the other arm of the other cell. So it's a disadvantage for IgG antibodies, while the IgM is better because of its pentameric structure. It is able to bridge the gap between the red blood cells and brings them together for agglutination. So to observe this type of reactions that happen between antibody and antigens in the red blood cells, we can do that using what we call the tube testing. What we call tube testing. Okay, tube testing, which can be performed in stages. Okay, can be performed in stages. We have immediate spin phase. Okay, during this process of antigen antibody interaction, we can bring about the process of antigen antibody interactions in red blood cells using tube test method. Okay, and under the tube test method, we can do it in various way or various stages, and each of the process or the stage we are using depends on what exactly we are looking for. We have the process of immediate spinning. Okay, here in tube testing, let's give a bit big of background, because in ABO tests, okay, in ABO testing, that's what we call the blood group typing. Okay, we use it to determine the presence of A, B, or other antigen, or none of them, the O blood group. Okay, but the ABO test can perform it together with the resource, or perform it separately. Okay, so that if performed together, they provide complementary information crucial for safe blood transfusion. Okay, because a mismatch in the ABO blood group or resource system can trigger severe immune reaction. So testing for both ABO and resource ensures compatibility on these levels and minimize the risk of transfusion reaction. Okay, guarantee safe blood transfusion. So in the process of cross-matching blood, we can do that by various methods, like we are talking about the tube testing. Now, the tube testing we are using We can perform either an immediate spin. Okay, we can take it up also and incubate at 37 degrees centigrade, or also perform indirect antiglobulin testing. Okay, depending on the antibody we are looking for, the type of antibody we are looking for, and the urgency of the required blood grouping. So to perform the tube testing, we say we can perform an immediate spin. Immediate spin meaning that we'll do like 2 to 5% of washed red blood cells for suspension, okay, and spin it, mix it with serum, okay, the serum of the patient. Then you spin washed red blood cells. You use 2 to 5% of washed red blood suspension, mix two of them together and spin for 30 seconds. This process is mostly used to detect mostly IgM. You can only detect IgM antibodies. Okay. So in order to detect other type of antibodies, we can take the tube testing method further, like incubating at 37 degrees centigrade is also a phase. In this phase, you can add what we call a potentiator, okay, and then incubate it at 37 degrees centigrade and spin. Depending on the potentiator we are using, if we have LIS, that is low strength, low ionic strength saline, you incubate for 10 to 15 minutes. If you're using PEG, the incubation time is 15 to 30 minutes. Okay, if you are using if there are no potentiators, that is no enhancers for the reaction to occur, the incubation can last up to 30 to 60 minutes if there are no potentiators added. So taking this phase further will enable some antibodies, maybe IgG, to be detected, that are IgG to be detected, because the potentiators or the enhancers that are added will encourage the reaction, agglutination reaction, to occur. We can also carry out tube testing in the form of indirect antiglobulin test. Okay, that is anti-human globulin phase. Okay, we wash above to remove unbound globulins. Okay, after the incubation period in the 37 degrees centigrade, we wash the incubated products to remove any unbound globulins, and then add the anti-human globulin and spin. Okay, add the anti-human globulin and then spin it. So this process of tube testing, we can be able to detect antibodies that are IgM. We take it further by incubation and addition of globulin, anti-human globulin, to be able to detect the IgG antibodies that are IgG, that are incomplete. So we use some potentiators to enhance the reaction so that it will be possible. Okay, there are also other methods alternative to tube testing. Okay, in some areas, in some localities, they have they can use column agglutination technology, which we call gel testing. Okay, they can use they can use other methods apart from the tube methods. Okay, so we have the alternatives to tube testing. Okay, so column agglutination technology. Okay, in this process, you have a gel, something like a gel inside the column. It's a newer method for ABO and rhesus blood tests, and it is more expensive, more rapid and efficient method. It's more efficient than the tube method. So to perform the gel method, a drop of the patient's blood is placed on the gel column. Okay, the gel contains antibodies to A and B antigens. Okay, the gel is then incubates at room temperature for 5 to 10 minutes. Okay, 5 to 10 minutes after the gel is examined for clumping. If the blood clumps with either antibody, it indicates that the corresponding antigen is present on the red blood cells. Like I said, the method is rapid and accurate for ABO blood grouping. Okay, it is often used in blood banks and maybe in emergency rooms. So red blood cell and plasma are added at the top of the tube, incubate spin. Okay. Okay. Like I said, the microtubes are filled with gel particles and anti-IgG. The anti-IgG grabs onto the IgG coated red blood cell and inhibits their migration through the gel. Okay. Gel particles separate red blood cell clusters physically, that is, inhibits agglutinate from migrating through the gel because a reaction has occurred, so they are bound at that specific place. If there's reaction, they don't go, they what we call eluates, we don't go down the capillary tube, they stay at the top position because a reaction has occurred, agglutination has occurred, and they can no more freely move down the gel. So the result is negative. Red blood cells form at the bottom of the microtube. Yes, if there are no agglutination, the red cell will move throughout the column down to the bottom of the gel. Okay. But if the reaction is positive, the red blood cell will stop in areas through the microtube. That is, it will stop in areas up where the reaction has taken place. Okay. It can be automated. Okay. So we said it's it gives more accurate result than the tube method. We also have ABO blood grouping or reactions can be performed as a solid phase red cell, can be also be performed in a solid phase. Okay, so this method is similar to what we observe during the process of ELISA, where we have microwells with antibodies, coated antibodies. And then we introduce the serum, the reaction interaction occurs with it, the antigen and the antibody, and the reaction continues. So in the same way, in ABO blood grouping, rhesus blood grouping, antibody binds to lyse or intact red blood cell antigens that are bound by manufacturer to the sides of the microwells. So these microwells are coated with the red blood cell antigens. Then the patient's serum is added, incubated, and washed. Then if it is positive, during the process of the incubation, the antigen antibody reaction takes place. Okay, remove the excess fluid and unbound globulin. If it is positive, antibody binds to test red blood cells. Okay, to indicator red blood cells coated with monoclonal anti-IgG attached to antibody on test red blood cells. We spin and interpret. For negative results, red blood cells in a button at bottom of microwells indicate cells didn't bind to the test red blood cells. Okay, so if we observe red blood cells in the bottom of the microwells, it means that they didn't bind to the test red blood cells. It is positive when the red blood cells spread in a carpet all along the microwells, indicating that red cells did bind to test red blood cells. It can also be automated, similar to sensitivity to PEG tube testings. Okay, so these are some of the processes by which we can evaluate the interaction, study the interaction between the red cell and the red cell antigens using precisely agglutination. Agglutination testing, you can study it using the tube testing or the gel method or the solid phase process. So we have the antiglobulin tests. We have the antiglobulin tests. It is we use to demonstrate in vitro red blood cell coating with antibody or complements. What does that mean? Okay, means that if in any situation or condition that a patient's red blood cell, either in vitro or in vitro, that is in the body or outside of the body, have been sensitized. Okay, we use this test to study red cell sensitization, meaning attachment of antibodies to red cells, either inside of the body or in the test tube. Okay, indirect, we have the indirect and direct antiglobulin test. In the indirect test, we use the indirect antiglobulin test to demonstrate in vitro red blood cell coating with antibody and or complements. In this process, it means that we are screening a patient for antibodies we are suspecting that the patient have developed. So we get the serum of the patient and incubate it with known red cell antigens so that we can detect the antibody in the patient's plasma. Or we have another type of the antiglobulin test, which we call the direct antiglobulin test. Here, we use this process to test red cells from patients, okay, red cells from patients who have been sensitized in vivo. Okay, there is a reaction between the patient red cell antigen and antibodies in vivo inside the body. So we use the red cells of the patients, wash it, then mix it with anti-human globulin to demonstrate in vivo red blood cell coating with antibody or complements. So the antiglobulin test, also known as the Coombs test, is used to identify red blood cell sensitization. If it is indirect, it means that the sensitization will happen in vitro. That is, we are screening for antibody in the patient. If it is direct, it means that the red cells in the patient are already been sensitized. Okay, the red cells are already been sensitized. Okay, so whether in vitro or in vivo, we use this test to evaluate such incidents. Okay, the indirect antiglobulin test, okay, has some, we can use it in certain situations, that is to study unknown antibody check, unknown antibody. Okay, maybe a patient is being sensitized, has an immune reactions, okay, but we don't know exactly the antibody that causing the immune reaction, if it's an antibody. So we can use the indirect antiglobulin test to check for unknown antibodies. Here we use red blood cells with known antigen profile, okay, as an antibody screen. Yes, then we also use the indirect antiglobulin test to check for unknown red blood cell antigen check. Here we use serum with known antibody specificity, as in red blood cell antigen testing. Or we can also use it, can be used to check for an unknown antigen or an unknown antibody, as in the cross match procedure. That is, in cross-matching blood, if we are cross-matching blood, what this statement is trying to emphasize is that in the process of cross-matching blood, we can be looking for unknown antigen. We can also be looking for known antigen. Okay, so we perform both direct and indirect antiglobulin test or Coombs test. So performing both direct and indirect antiglobulin test means that we are looking for unknown antibody and we are also looking for unknown antigen. So um, IgG sensitized red blood cells can serve as controls. Thus, we use it for controls. There are antibodies that have been sensitized with IgG. So when we are performing Coombs test, we need also to add the serve as control. Okay, so that if we don't get the reaction we are supposed to get during the process of screening the patient with this positive control IgG sensitized red cells, it shows us that our something may be wrong with our reaction. So it's just a positive control during the process of antiglobulin testing. We need to add IgG sensitized red blood cells and to okay, so we add IgG coated red cells to AAG cell mixture. If it is negative, then bad AAG or no AAG added, or there may be other errors. So IgG sensitized red blood cells are used in as positive control during Coombs tests. That's the purpose of it, because it's already sensitized with IgG. So it's expected that during the process of antiglobulin testing, that it's this particular control must come down with negative result, positive results. So if it does not come down with positive result, it means that the AHG, the anti-human globulin reagent we are using, is bad, or that we did not add it, or maybe we did not add the serum, that there's a problem with the process. So we need to repeat it. So in any laboratory evaluation anyway, you must have your positive control and if possible negative control. So to control the process of antiglobulin testing or Coombs testing, we used IgG sensitized red blood cell. Okay, so specificity possibilities for antiglobulins. Okay, the antiglobulins that are used have their specificities, like the anti-IgG. Okay, anti-IgG as complement 3D. They are polyspecific. Okay, the most common to start. Okay, so this antiglobulin that is have a IgG Ig 3 complement, they are polyspecific. Okay, they can detect red cell coated with either IgG or complement 3D. So the antiglobulin kits have some specificities. Okay, the IgG and CD3, that is complement 3D. Okay, they may also detect other immunoglobulins because the IgG detects light chains too. Okay, so we have some antiglobulin that are IgG, anti-IgG, heavy chain specific. Both they can detect IgG coated red cells, anti-IgG used for PEG, gel, and solid phase tests. They can be used. We have the antiglobulin C3D minus C3D. It can also detect either of they are both complements. It's most useful in evaluating IgM-related hemolysis, cold agglutinin disease. So some of this IgG, that commercial IgG, have their specificities, which should be considered at the time of purchase. So the anti-IgG C3d is polyspecific. Okay, polyspecific, the most commonly used, the polyspecific antiglobulin. So the reaction between red blood cells and their antibodies also show what we called dosage. Okay, dosage, meaning that some antibodies react more strongly with red blood cell antigens that have homozygous gene expression. Okay, we talked about the dominance of the ABO blood group, but sometimes there is variation in the expression of these ABO blood groups. So this expression variation also contributes to ABO blood grouping. Okay, for example, imagine a hypothetical anti-W. Okay, patient is homozygous for this antibody. Then patient 2 genotype is heterozygous WY for W antigen. So if anti W show dosage, it will react stronger with patients one's red blood cell, as we can see. Okay, patients with the homozygous gene can react stronger, show more agglutination than patients with the heterozygous gene if the genotype we're considering shows dosage. Okay, this dosage occur mostly in some other blood groups like the KID, the Duffy, the Rhesus, and MNS system. We'll be looking at blood groups and other blood groups later. So sometimes the ABO blood group system and other blood group systems show dosage effect in their reaction. You see some persons with stronger reaction. So enzymes also affect ABO blood group reactions. There are some enzymes that are proteolytic enzymes, it's the ficin and papain. They cleave red blood cell surface glycoprotein and can strengthen reactions, okay, by enhancing antigen expression or allowing antibodies to bind better to previously shielded antigens. So sometimes enzymes, like we see here, the proteolytic enzymes cleaves red blood cell surface glycoproteins and can strengthen reactions. Okay, sometimes they remove some obstructions on the red blood cell surface, enhancing the reaction of the antigen and antibodies. Okay, when they cleave some of these proteins, then they cleave some of these glycoproteins, they expose the antigenic surface of the red blood cell and then enhance the interaction between the antigen and the antibodies. Sometimes this enzyme may also directly destroy other antigens that are also contributing to inhibiting the reactions with the specific antigen and antibodies. Some of the enzymes are useful in antibody identification to confirm or refute a particular antigen as the target of an antibody, as you can see in this table below. Okay. So these systems are enhanced by enzymes. Okay. These enzymes decrease their reactions. The Kell system, the Diego system are unaffected by enzyme activity. the MNS system, the Duffy, the Lutheran system, their interactions, the antigen and antibody are decreased by enzymes. But for the ABOH system, Lewis system, I system, P system, resource system, and the KIT system, their interactions with their specific antigen and antibody are being enhanced by enzymatic reactions like the proteolytic enzyme, the ficin, the papain we talked about earlier. So another reaction that happens between ABO blood group and antigen and antibody is what we call neutralization. Okay, when mixed with a red cell antibody, certain substances inhibit that antibody activity against test red cells. Okay, like take for example, we have in this first row ABO. ABO blood group is being mixed with saliva from secretor. Okay, it tends to inhibit the interaction between ABO blood group antigen and their corresponding antibodies. Also, the Lewis system is affected by saliva, the PI system affected by hydatid cyst fluid, okay, the Chido Rogers are being affected by serum. So these substances can neutralize the interaction between blood cell antigens and their antibodies. We'll call neutralization. So we have to bear these things in mind because it's necessary as you carry on ABO blood group um, study. Maybe you are trying to screen for ABO blood group in saliva. You should be aware that saliva inhibits red blood cell antigen antibody interactions, and then now know the steps to take so that you'll be able to get accurate results. Also, we have some lectins. These are seed plant extracts. They have ABO, that's ABO antigens. Okay, they are useful in polyagglutination, may be commercial or homemade, may be made commercially or homemade. So these lectins or seed extracts or plant extracts are very good in agglutination. They sometimes help to identify some subgroup blood types. They are specific for A1 blood group like Dolichos Ulex Europeus. They are specific. You know, when we talk about the ABO blood group, we have subtypes of each blood groups where we have blood group A, we have A1, A2, B group, we have other subgroups. So some of these lectins are specific for some of these blood group subtypes, and when we use them we'll be able to identify these blood subtypes like the Dolichos okay, we can use it to is specific for A1, an individual that is A1. We can use it to identify an individual that is A1. And these lectins are either seed or plant extract, extracted from plants or seed, but they are specific. They can identify some human blood subtypes. Okay, just like this table is telling us that Dolichos can use it to identify an individual that is blood group A1. Okay, Ulex Europeus, you can use it to identify an individual is specific for H antigens and so on. Okay. Okay. So, like I said, we are going to stop here. The next lecture said it's a very brief lecture. The next lecture will talk about the ABO blood group and other blood group systems. Okay, so like I said,